Hello, my name is Nara Souza and I am a biologist with a master's degree in aquatic biology. I have been working with algae and cyanobacteria for over 8 years, and today I would like to talk to you a little bit about climate change and harmful algal blooms. Here we have examples of some groups of algae that are present in marine and freshwater environments. We have green algae, diatoms, dinoflagellates, red algae, haprophytes, and cyanobacteria. Some of these groups have members that are capable of producing toxins like diatoms, dinoflagellates, haprophytes, euglenoids, and cyanobacteria. I also would like to say that these organisms are natural occurring and very important to these ecosystems, and many have several properties of health benefits. The problem is when things get out of balance and they bloom. One important factor that influences bloom is nutrient. Although there are numerous benefits to these organisms, there are many compounds that cyanobacteria and algae can produce that have harmful effects. Here we have some of the common toxins. Microcystins and nodularins are produced by several species of cyanobacteria and can inhibit protein phosphatase. A pronounced sign of acute intoxication by these toxins in mammals is the destruction of liver macrostructure by the deformation of the cytoskeleton in the hepatocytes. Microcystins have been found to accumulate in multiple organs and tissues in mammals and fish, such as heart, liver, lung, brain, and kidney. Cylindrospermopsin, also produced by cyanobacteria, has been shown to be an inhibitor of protein translation within the eukaryotic cell. Enatoxin A mimics the neurotransmitter acetylcholine and will bind to acetylcholine receptors where it cannot be removed and results in continued nerve impulses. Guanetoxin, formerly known as enatoxin AS, is a naturally occurring organophosphate and, like organophosphate pesticides, inhibits acetylcholine esterase, an essential enzyme for removing acetylcholine from nerves. A potent neurotoxin, saxitoxin, binds to voltage-gated sodium channels and results in continued nerve impulses. Similar to saxitoxin, brevetoxin is also a sodium channel blocker and is volatile, resulting in becoming airborne and affecting breathing. This toxin is produced by the dinoflagellate Carenia brevis and is commonly known as red tides. Ciguatoxins, produced by the marine dinoflagellate Gombiardiscus toxicus, is a neurotoxin that acts by opening the sodium channel. You can get ciguatera fish poisoning by the consumption of ciguatoxin-contaminated reef fish. Domoic acid, produced by the diatoms Pseudonychia, is a neurotoxin that is excitotoxic of glutamate receptors and results in a neural depolarization. It is involved in the amnesic shellfish poisoning through the ingestion of contaminated seafood. BMAA, produced by cyanobacteria, has been shown to act like glutamate but can also be incorporated into proteins during protein synthesis. These toxins have a wide range of effects and can impact a number of organs and systems in humans and animals. Some of the main areas that may be affected are the liver, the nervous systems, and the gastrointestinal tract. Other organs such as the skin and kidney as well. In high enough doses, many of these toxins are sufficient to kill. The effects of these toxins can be acute, with some acting in a very short time. Such effects can include rashes, vomiting, fever, as well as respiratory arrest, liver failure, and death. However, increasingly, we are beginning to understand that long-term exposure to these compounds may cause chronic health issues of the nervous system through the exposure of BMAA and domoic acid, and microcystins, nodularis, and aplasia toxins have been implicated as cancer and tumor promoters. So how do we get exposed to these toxins? Of the routes possible, the major ones are oral, through the ingestion of contaminated water, contaminated food such as fish and shellfish, contaminated crops, and dietary supplements, some of which have been shown to contain toxins. Other routes include inhalation of aerosol and dust produced during storms that may contain algae and toxins. Finally, recreational exposure may also be an issue through water sports such as sailing, windsurfing, and water skiing, in addition to bathing in such contaminated waters. Now, how can climate change potentiate algal blooms? Increasing temperatures will favor blooms, especially cyanobacterial ones. Under such conditions, they may outcompete other species of algae. These higher temperatures may also increase the frequency and duration of blooms. Other effects of increasing temperature can include stratification of water bodies that could also favor algal blooms. As climate change affects temperature, this is also likely to expand the geographical range of some species. As for example, Cylindrospermopsis rossiboskii, although thought to be a cosmopolitan species, some researchers think that climate change with increasing of temperature have benefited this species' geographical expansion. Another consequence of climate change may be the occurrence of droughts, which may result in the loss or shrinkage of water bodies. This will have the effect of increasing nutrients, favoring algal blooms, in addition to increasing residence times, which is also a favorable factor for algal blooms. With the possible result of shortage of water supplies, humans and animals have then a higher risk of exposure to such contaminated waters. Another issue of climate change is the increasing likelihood of severe weather events. This will probably cause superstorms to result in flood and storm surges, increasing nutrient runoff into water bodies. Such storms can also lead to aerosol production and dust storms containing inhalable toxins. As rainfall is affected, along with droughts, this could also lead to desertification, with a loss of soils and cropland, releasing nutrients to water bodies. This loss of soil could likely become airborne, transporting algae, nutrients, and toxins. Although we are observing the effects of climate change, there are still things that we can do. One of the major ways of controlling algal blooms is through nutrient control, either at the source or through the restoration of wetlands that can act as filters to water. Monitoring, it's a tool that can help us understand and know what kind and where toxins are present and whether our efforts to control blooms are working. It can also prevent exposures. Creation of legislation for drinking and recreational water to help drive our efforts to control blooms and to ensure clean water. Another need is research, as this will be an important tool to give us solutions for us to deal with and learn more about climate change and harmful algal blooms. Thank you very much for your attention.